Good morning. Welcome to a briefing by Johns Hopkins University in Medicine about mRNA. We'll be hearing from three researchers who will talk about how the technology works and its potential to treat and prevent disease. There will be an opportunity to ask questions at the end, but please place questions as you have them in the Q&A box. Our first speaker will be Jeff Coller, the Bloomberg Distinguished Professor of RNA Biology and Therapeutics, who has studied mRNA for more than 30 years. He'll discuss the history and development of mRNA technology and why he thinks it has the potential to revolutionize the treatment for certain diseases.
Thank you. Share my slides. Good morning, everybody. Again, my name is Jeff Coller. I'm going to tell you a little bit about mRNA medicines, which we're considering to be a new pillar of medicine and a biomedical revolution. So just to start out, what is mRNA? So mRNA stands for Messenger RNA, and mRNA is the genetic emissary of DNA. And so every gene in your body will encode an mRNA. And the purpose of that mRNA is to transmit this genetic information out of the nucleus where DNA lives and into the cytoplasm, where it then binds to a protein RNA complex called a ribosome that then makes proteins. And then proteins do the work within the cell. One of the benefits of mRNA in use as genetic information is that it is regulated and it can be turned off and on at the level of DNA. And in the cytoplasm, what's really important from a medical standpoint is that mRNA is very unstable. It is destroyed over time in a very programmed manner. And so it dissipates. And this information then is lost. And so that becomes critical because it's cleared from the body and becomes very safe for the use in a medical standpoint. And so an mRNA medicine is a synthetic mRNA, a piece of genetic information that we introduce into the cell in combination with a lipid nanoparticle that then goes into the cytoplasm and then makes a protein of our design. So mRNA now represents the next and fourth pillar in pharmaceutical intervention into human health, right alongside small molecules, biologics, and cell and gene therapies. Many people think that mRNA only came on the scene in the year 2020, but it has a 65-year history. mRNAs were first discovered in the 1960s, and their use as medicine was explored heavily in the early 2000s. And in fact, the first in-human tests of mRNA in the cancer space occurred in the year 2013. And so we know a lot about the use of mRNA in the human body, its safety, and its efficacy. But of course, mRNA became a household word in the year 2020 with the advent of the COVID-19 pandemic. And we all remember what happened during this time. And it was really the leadership of President Trump that led to Project Operation Warp Speed on May 15th, where it was officially announced, where we began to harness the power of mRNA to fight this pandemic. In addition to using mRNA to treat infectious diseases like COVID and other viruses, mRNAs are now being explored to do other intervention, including the treatment of cancer, metabolic and genetic disorders, cardiovascular diseases, and other rare genetic disorders. And so it goes well beyond the use of just in vaccines. A particular example of that is the treatment of pancreatic cancer. So a recent report in Nature demonstrated that a personalized mRNA vaccine, similar to what was described at the White House Stargate announcement by Larry Ellison, generated a strong anti-tumor immune response for half the pancreatic cancer participants. Similar results are being shown in other solid-state tumor cancers, like melanoma, as well as rare diseases like cystic fibrosis and Duchenne's muscular dystrophy. But despite the promise of this technology, and celebrating and in continuing the investment in this science that really saved us, we're risking squandering our leadership in the mRNA space in the 21st century. As NIH implements deep cuts to both the headcount of personnel that are working there, as well as funding, NIH researchers across the United States are being targeted for projects involving mRNA vaccines. And based on what we've seen in other research areas, the next steps could be from the administration to halt funding for projects that are developing these breakthrough mRNA therapeutics that are treating cancer and other lethal disorders. In addition, the huge cuts and experienced staff at the FDA, including CBER Director Peter Marks, whose vision provided the regulatory leadership necessary to make the United States a center of mRNA innovation, leaves patients, scientists, and innovators with significant questions as to the future of the United States in the leadership within the biomedical sector. In addition, we are seeing that this technology is poorly misunderstood by the community. There are a number of states that have on the docket bills that would either ban or restrict the usage of mRNA in treating various disorders. But despite this, the science is really clear. mRNA medicines are extremely safe. There are publications that have studied well over 100 million vaccinated individuals that were treated with the COVID vaccines, and the number of adverse events that are seen is similar to what is seen with other vaccines. And in addition, it's been shown that the Project Operation Warp Speed saved well over 20 million lives. And this should really be celebrated as an achievement of the Trump administration's first term. And indeed, on the second day of his presidency, President Trump seemed poised to build on his first term success when he announced Stargate, which was a $500 billion venture to advance artificial intelligence within the United States. And during that White House press conference, Oracle Chairman Larry Ellison stood next to President Trump and emphasized an opportunity to use AI to speed personalized mRNA treatments for patients living with cancer, bringing hope to millions of Americans whose families live and are upended by cancer every year. In addition, this technology is expanding at a breakneck speed. It will continue to develop new vaccines over the next decade. But what you can see is that it's also expanding into other areas that I've already mentioned, including treatments of cancer and rare genetic disorders. And it really represents an over $30 billion industry by the year 2030. And so it's not only a biomedical revolution, it's an economic opportunity for the United States. Currently, there are over 700 assets in preclinical development in the mRNA space, most of these being housed at companies that reside within the United States. And 267 of these assets are currently in clinical development. I'm one of the founders of an organization called the Alliance for mRNA Medicines, and this organization is done a number of studies on the impact of potential federal cuts to mRNA research on this industry. Now, the Alliance for mRNA Medicines, it has over 75 plus groups, including Johns Hopkins and other academic centers, and many of these organizations shown here. In March of 2025, we conducted a survey, and this is important to note that this survey was done before the changes at the FDA, where over 100 leadership professionals, life science professionals, responded to our survey about how they felt federal cuts would influence the mRNA space. And the results of that were really quite clear. Over half of all of the leaders that we surveyed said that these cuts would delay initiatives in the mRNA space, and that they would have to seek alternative funding sources and may indeed have to terminate specific programs. And perhaps most devastating is that about 30% of them would consider relocating their operations to other countries if the United States pulled away from mRNA technologies. Now, currently, about 65% of all jobs in this space are housed within the United States. This is over 150,000 scientists and workers in this workforce. And we anticipate that there will be a significant reduction in this workforce if federal funding to mRNA research is cut. Over half of all of our surveyed participants reported that they are already experiencing a direct impact of the policy changes that have come forth from the administration in the last few months. These impacts include reduction in scope for projects, budget reductions, delayed capital investment, partnership terminations, job loss, and hiring freezes, as well as potential relocation of projects and their companies to other shores. Indeed, in conversations with our members, strategic partnerships have been lost because NIH funding has been cut, and companies are already looking overseas to turn their manufacturing operations into other countries. Currently, the way the market stands in the mRNA space is about half of all mRNA companies are housed within the United States. But by 2023, we expect that the rest of the world will really catch up. And if we reduce our investment into the mRNA space, then these other countries will definitely dominate, and America will lose its leadership. And with the threat of indiscriminate cuts to mRNA programs, governments around the world, like China and the European Union, Australia, and Brazil, are actually not retreating from this technology. They're doubling down. Efforts to undermine mRNA technology, as we're seeing by recent actions at the NIH and FDA, will force these potential cures, therapies, and related manufacturing jobs to go overseas. Europe and China will lead innovation to treat cancer and rare disease, and the United States will not. This is not a partisan issue. This is a bipartisan issue. This is a report that was released in February of 2025 by the Texas Conservative Coalition, which spotlighted the mRNA technology. The Texas Conservative Coalition was founded in 1995 and is a group of conservative leaders that help advise policy decisions based on a conservative agenda. And their recommendations in February basically are very logical, consistent with the messaging that I'm saying, that mRNA should be heavily invested into as it is a life-saving technology. We should fund mRNA vaccines and therapeutics because this funding is absolutely critical. It's critical for reorganizing the potential of mRNA technology to address unmet medical needs, for preserving the momentum of the pipeline that's already existing, to bridge funding gaps that will occur, to ensure a sustainable mRNA medicines workforce so that we can lead into the future, as well as maintain global health and reap the economic benefits of dominating in this space. In addition, the more broad implications of NIH cuts to federal funding is that America's brightest minds will walk away. This is an excerpt from a New York Times article, which basically shows that 27% of the world's total research and development activity is done in the United States. And a close, very close second to that is China. And it is estimated that every dollar of NIH funding produces at least $2.50 of economic activity. And so we would argue that the mRNA space and all biomedical space should be continued to be invested in by the federal government. Thank you.
Thank you, Jeff. We had a couple of questions in the meantime about whether the slides will be available. Hit me up by email afterwards, and I will send whoever wants the slides, and everyone who's registered will get a recording. Next up, we'll hear from Elizabeth Jaffee, an oncologist focused on immune-based therapies for pancreatic and breast cancers, and Deputy Director of the Sidney Kimmel Comprehensive Cancer Center. She will discuss the promise of mRNA technology for cancer treatment and prevention.
Thank you for the introduction. So I wanted to put mRNA vaccines in the greater context of where we are in cancer therapy. The investment by the NIH of the past 3 to 4 decades in understanding cancer biology has really led to a time where we are just in an unprecedented period where we are developing treatments that are actually saving lives. And the immune system being able to activate the immune system is really the biggest advance we've had in this field since cancer was ever diagnosed, hundreds of years ago. Well, what are these drugs that are so important? They're the immune checkpoint agents, immune modulators. So we've heard about anti-PD-1, anti-CTLA-4. They're approved for cancers like lung cancer and colon cancer and other cancers that previously were deadly cancers. And patients are living longer, much longer. Some are cured of their diseases because of these treatments. Well, these treatments, again, due to a significant NIH investment over many decades, have allowed 20% of all advanced cancers that were deadly to become chronic diseases like diabetes, okay, where you can live a good life as long as you continue to control the disease with the therapy. The problem is 80% of those patients, 80% of our patients still don't respond to these. And we've figured out, again, due to the investments at the NIH, the new technologies, we're in a technological revolution in cancer research. And again, investments over the past several decades by the NIH have led to new technologies that allow us to take a piece of a patient's tumor at biopsy and study it and understand why are they resistant to immunotherapy? And what we're finding is that there is the patients who are resistant to these wonderful agents, these miraculous agents, they're resistant because the immune cells that they act on, called T cells, have not gotten into the cancers because they're not there. So 80% of cancers, just for some reason, don't alert the immune system to the fact that they are growing and progressing. Well, then comes mRNA vaccines. And actually, to be honest, we've been studying mRNA vaccines at a preclinical level prior to COVID. So we all saw as scientists, as immunologists, as physicians, that these mRNA vaccines would be very important for cancer patients and hopefully to prevent cancer in the future. So why are they important? Because we know that these agents have the ability to activate, even in patients with cancer, an immune response, a T cell response that can get into the cancer and can collaborate with these immune agents that have been so successful in 20% of bad cancers. So that's why we're very excited about this. And this is why we cannot allow research to go away in this field. We have the opportunity to make a big difference in many patients' lives. Well, why do they work so well? Well, they work so well because all we have to do is take a small biopsy again of a patient's tumor, which is standard for most patients with cancer. And we now have the technologies to sequence them, to identify proteins that are expressed specifically by the cancer, but not by the normal tissue. So again, that's important because if it's cancer specific, we're activating an immune response just to see the cancer. Well, that's what mRNA allows us to do. It allows us to identify those proteins. They could be 5 proteins, they could be 20 proteins. mRNAs are one of the few ways in which we can actually incorporate these proteins so that we can then make a vaccine, give an injection under the skin, and activate a T cell response. And we've seen this. You heard from Jeff about pancreatic cancer, disease that I treat. These patients who have received this vaccine normally would have died within 2 years. It's a small study, the first study, but patients are living without the cancer coming back because of this mRNA vaccine. So now it's in a second study comparing no vaccine versus vaccine to determine the true value of the mRNA vaccine for these patients. But again, there's many different clinical trials that are ongoing, and the government helps to support these clinical trials early on to make sure they're done correctly by the experts in the field before industry is able to take them on to further development. And so we have studies now in lung cancer and pancreatic cancer. These are bad cancers that people are dying of, breast cancer, you name it. This vaccine, an mRNA vaccine, is so easy to make for patients, and it's patient specific. And so this is the only way we can see moving forward to make these immune agents even more available and worthy of having an effect on patient cancers over the next 5 to 10 years. I really see that if we can push this vaccine method forward, we will definitely have an impact in cancer that's going to be unexpected, but almost miraculous over the next 5 to 10 years. Now, there's another area that these vaccines can help in, and that is in high-risk patients who know they have genes that are making them susceptible to bad cancers like pancreatic cancer, like breast cancer, like prostate cancer, like all of the different kinds of cancers that you can imagine. In addition, they have family histories. There's many people who have many family members who have gotten bad cancers and have died of cancers. Well, now we're able to understand, again, what are the early proteins that change in a normal cell that help the normal cell become a cancer. These mRNAs, which have been given to hundreds of millions of people, these mRNAs can now be used in the same way we gave it to people who are healthy, to those people who are at high risk for different kinds of cancer, hoping that we will prevent them from developing those bad cancers. I mean, this was a dream of mine when I got started in the field 3 or 4 decades ago. Now it's actually almost a reality. So to lose this funding that's supporting this work really will be detrimental to many patients currently with cancer and future patients.
Thank you so much, Dr. Jaffee. A reminder for anyone out there listening who has questions, go ahead and put them into the Q&A box and we will get to them after everybody's finished. Our next speaker is Jordan Green, a biomedical engineer who develops new biotechnology to improve the delivery system of mRNA. He'll talk about how the technology can be used to treat rare genetic diseases, autoimmune diseases, and other conditions.
All right, thank you. It's a pleasure to be with everyone today to talk about mRNA delivery and how it's enabling the future genetic medicines. And so an important aspect in this field to really appreciate is that mRNA delivery is a general tool for a new paradigm of medicine. So it's much broader than vaccines that can be used in a preventative way. It can be used therapeutically in many ways as well, and really touches just about every area of medicine and human health. It can enable very precise and controllable expression of desired genes. And it's the expression of genes that ultimately really lead to all of our different diseases. And so this is a technology that can then be utilized to address virtually all human diseases. As Jeff talked about, in the standard way of using mRNA, it's actually just a very short, defined expression, and then it degrades away. And so this can be used with vaccines and some other treatment approaches where we only need expression for a short time to kickstart the immune system, for example, or to provide a certain kind of healing or a certain type of reprogramming that then the cells can take over and provide this healing. In other ways, though, we can use mRNA to express certain proteins that can make a long-lasting change. And so that's through gene editing. And so the mRNA can encode the machinery that'll just be there a short time, but can then make an effect that could be long-lasting. So there could be a one-time dose that leads to a cure potentially. And this could both be used to turn on a gene as well as turn off a gene. But the big difference now is that we're looking at medicine in a molecular way where it's very precise and going to really the root of the disease. What has been accomplished with mRNA delivery has been extraordinary. And in many ways, it's like designing a rocket ship to Mars, and that it should really be celebrated as such. It was tremendous science and engineering for this fantastic voyage of what the mRNA is doing, but instead of going out to outer space, is going to inner space, this fantastic voyage through our bodies to go to the right cells, to deliver the right instructions, to then create a whole paradigm of healing in the body. And so for this to work, the nanoparticle, which was the vehicle used instead of a rocket ship, it's a nanoparticle, and I'll talk about that in the next slide. It has to overcome many different barriers on this journey, and that they've been designed now to do this very safely and effectively. In addition, the next generation rocket ships are also being designed to be even more broadly accessible so they can really help heal the world in a way that is able to not be very expensive and to continue to be very safe and effective. So what is a nanoparticle? It's a very small particle. And on a length scale, if we think of the world and then a globe of the world, that difference in size is about 10 million. And so just as you shrink from planet Earth down to a globe of the Earth, to shrink from the globe down to a nanoparticle, it's that same difference of 10 million in size. So that's how small they are, about 1/1000th the width of a human hair. And yet this little particle has to do so much in order to do its job and deliver that mRNA safely. And a lot of these are designed so they degrade in water very safely using biomaterials that are very safe, and that they can carry different drug cargoes, especially including the mRNA. What are these particles made of? The traditional particles are lipid nanoparticles. So that's little balls of oil and fat, basically, that are able to protect mRNA, which is a very sensitive molecule. Again, in the body, it naturally degrades very quickly. So it needs to be protected. And so these little balls of oil and fat are able to do that and protect the mRNA until it reaches the cells. Other kinds of particles that are also being developed, including here at Hopkins, are polymeric nanoparticles, and that those have certain advantages as well in terms of their manufacturability and their targeting and their biodegradation. With a polymer, it's something not unlike biodegradable sutures or biodegradable stitches, where they don't have to be removed after because they are able to naturally degrade in water in the body. And so there's very similar types of polymers that are used in medicine that can make these particles very effective for mRNA delivery. And so how do these particles work, whether it's the lipid nanoparticles or the polymeric particles? They have to go through these different steps, and this is like that little rocket ship in the body. So it has to protect the mRNA so that it doesn't get degraded. And then it has to lead to internalization into the cell. And so the cell has to be induced to eat up the particle, which is like us eating food. And just like when we eat food, we digest what we eat. The cell inside its little stomach, this endosome, it would normally degrade these particles, degrade the mRNA, and nothing would happen. And so the little rocket ships, the nanoparticles, have to be designed to escape this endosome and get in the cytoplasm, which is where they work, as Jeff showed how that works in the cytoplasm, for them to make protein and have their effects. And the particles also then have to safely release the cargo and then degrade and get out of the way. And so this is what these rocket ships can do. And so it's a really powerful technology that's much broader than vaccines. And now we're at the precipice of this new era in medicine where it's genetic molecular medicine. And so one way this mRNA medicine can be used is that the body can become a protein drug factory. So instead of having very expensive proteins that are made recombinantly that need to be injected in the body over and over again every day, someone's whole life, we could instead have mRNA induce this expression of these protein drugs within the body, within the patient. They're their own factory for producing the protein that they need. And so this means less frequent injections and less costs compared to conventional protein drugs. It also can lead to potentially improved outcomes because there's local production and sustained production instead of the big peaks and troughs of having injections. And again, it's very safe. And this can be used in all sorts of ways that proteins are used in medicine: hormone replacement therapy, enzyme replacement, to treat different degenerative diseases. Again, the idea is we give the instructions to the body on how it can produce its own medicines to heal itself. And so this is what mRNA medicine allows. Another really exciting area with mRNA medicine is in immunoengineering. And this is the ability of those mRNA instructions to teach the immune system how to do its job better, or how to correct itself if it's doing its job poorly. And so we want to turn the immune system on, for example, in cancer. And we heard a lot of that from Liz about these dramatic effects that are seen in the clinic with immunotherapy for cancer, that if we can train the immune system on how to better recognize cancer from healthy cells, it can do a wonderful job eliminating the cancer in a much safer way than chemotherapy or radiation or other conventional approaches. And so this can work across virtually all cancer types and melanoma, prostate, lung, liver, pancreas, and more. And then there's different approaches. So there's the cancer vaccine approaches we heard about with mRNA, but there's other approaches too. So we can have in vivo CAR T, where cellular therapies that are right now also looking very exciting for certain kinds of cancers. The cells are taken out of the body, engineered outside the body, and then put back in. And that costs hundreds of thousands of dollars. In the future, what we'll be able to do is use an off-the-shelf particle that then is injected systemically, will program your own immune cell, those T cells, in vivo to then express the factors it needs to fight cancer better. And this could be done off the shelf in a very inexpensive manner, in a very broadly accessible manner. And other approaches with cancer also look at reprogramming the immune microenvironment in a way that the treatment might not even need to be personalized to the individual. There could just be a general particle that can reprogram the immune environment. So whatever the cancer is presenting as its antigens, that's what the immune system will learn how to detect and then treat and remove. In infectious diseases, this is, of course, also very helpful and can work across a range of infectious diseases much broader than COVID. And there's clinical trials to look at it in many other infectious diseases. One of the advantages is the speed that these can be produced, as well as their safety and efficacy. There's also tremendous opportunity with turning the immune system off when it's gone awry. And so this is in different autoimmune diseases like type 1 diabetes, multiple sclerosis, Crohn's and colitis. mRNA can teach the immune system how to recognize itself, healthy cells, and no longer attack itself. And that's what happens in these autoimmune diseases, is that the immune system is actually attacking itself. And so we can teach the immune system to stop doing that. And in this way, mRNA could be a potential cure for these autoimmune diseases right now that are so tragic. And then another area of mRNA medicine is that it can enable genetic editing. And this is where, instead of thinking of a short-term expression of the change, that the short-term expression will lead to a long-term change on the molecular scale with gene editing. And this goes to fix a genetic disease at its source. And so a patient, instead of having to take pills every day to treat the symptoms of the disease, they can now potentially have a one-time injection that cures the disease. And we're seeing this in clinical trials that this can be very safe and effective on rare diseases that don't really have other ways that they can be treated. And it's very exciting to the field. And the same kind of approach could also be used in more common diseases that are acquired over time, where having a protein turned on or off can essentially cure the disease. And so it's very exciting. These are how the medicines are made. The sequence can be designed based in a personalized medicine way or in a more generic way. And then the mRNA molecule can be produced and purified. And then it's essentially just mixed with either these lipid molecules or the polymer molecules, and it'll self-assemble into a nanoparticle. And then that's what it is. So the rocket ship builds around that cargo, and it doesn't matter what the sequence of that cargo is. And that's what makes this therapeutic approach and the manufacturing so much more streamlined than many other medicines, is because that same rocket ship can be used with any kind of mRNA sequence, any kind of passenger you want to put in it, and then it can induce the healing. And so there's a really bright future ahead with mRNA nanoparticle medicines that are increasingly able to better target specific cells and tissues to then enable in the near future this well-defined and safe genetic surgery to treat and even cure many diseases at their fundamental genetic level. And that increasingly the manufacturing is getting more streamlined and inexpensive, and then the formulations are getting more stable and able for very broad accessibility. So putting this all together, it's a really exciting time. And we certainly don't want to be slamming on the brakes when there's so many patients whose lives are at risk that we can help in the near future. And thank you so much for your attention, and we'd be happy to answer any questions.
Thank you, Jordan. We do have some questions, some that were submitted in advance and some during the presentations. One that we have, there is concern within the U.S. research community that mRNA research funding could be cut. What is your perspective on that? And is there a reason to worry about the fate of mRNA technology research in the US?
Yeah, I can take that question if you want. I mean, I think there is a huge concern. RFK Jr. has been a strong critic of mRNA. That's public knowledge. He's stated this. And then under his leadership, there have been emails sent out to the intramural program at NIH that is also public knowledge that have asked investigators to disclose they're working on mRNA vaccines and their collaborators and who their collaborators are. And this is a pattern that we've seen in the last few months, that there are actions that then get taken based on these internal memos and such. So we are concerned. The industry is concerned, and even if even if nothing directly happens, there's skittishness within the marketplace and the sector. And when you think about building a new company or new factory to make these medicines, and you may go overseas rather than do it within the United States. And so all of this uncertainty is something that creates potential volatility within the sector.
I'd like to just add one point as well, and that is that our patients who come to our clinics and are getting vaccines, they're very concerned. They ask us about this because they're benefiting. And the future of their future family members, they're worried, are going to have cancer, and hope that agents such as mRNA vaccines will be available to their children and grandchildren should they need it. So there's a lot of fear among patients with cancer.
I'd like to chime in on this as well. It's a very good question. And as I was describing, mRNA technology is poised to be revolutionary in the practice of medicine. And it's far-reaching beyond vaccines for infectious diseases, for which is found to be safe and effective and also allows a speed in development not possible for other vaccine methods. But the same mRNA vaccine and delivery technology is useful to treat and potentially cure almost every known disease because it enables genetic surgery, being able to precisely heal the body at the molecular genetic source of the disease, rather than the conventional approach of just treating the symptoms of the disease, which is how many medicines right now work. So, for example, type 1 diabetes. This is caused by the body's immune system attacking its own insulin-producing beta cells in the pancreas. To treat the disease symptoms, a patient has to frequently inject themselves with insulin and make up for the insulin not being produced. With mRNA technology, we could treat this at the source by reprogramming the immune system to protect and not harm the insulin-producing beta cells. This would essentially cure the disease. This would be wonderful for the patients and their families, and would also significantly reduce healthcare costs. Lifelong diseases that require constant drug dosing could instead become a one-and-done cure. And this is far-reaching, from rare genetic disorders to common forms of cancer. And so these types of medicines truly are a revolution. And the question is America going to continue to be a world leader in biomedical research and medicine? Are we going to continue to create the cures that save the lives of millions of Americans, create high-paying jobs in biotech and in medicine? Or are we going to cede our preeminence to someone else? And we're really at this very important decision point as a country.
Thank you. We have another question about state level bills, which I think Jeff had mentioned in his presentation. What are your thoughts on the state level bills where concerns over mRNA could potentially become policy?
These are very disconcerting because they're based not on scientific evidence. They're based on politicalization of the fears of the technology, which are simply not based in the hard evidence. And the problem that we foresee is that any of these policies turn into law, then they could follow to the federal level as well. And so it's very important to continue to educate and speak to people in these areas, as well as our elected representatives, to speak to the power of the technology, the promise of the technology, the economic impact of the technology if it were to be restricted, as well as what Jordan said is, do we really want to be leaders in the world in the biotech space? in helping patients and saving lives. And the United States has had a long, rich history here. And human beings have benefited from the investment of the American citizens into the National Institute of Health. And these sort of policies are very scary, quite frankly.
We have a technical question. How does mRNA compare to gene editing technology like CRISPR? Can it be used for the same things in the same way?
I can answer that, and maybe Jordan can too. But one of the beautiful things about mRNA, and this is something my lab studied for 30 years, is that it's unstable. It's like a drug. It just gets cleared from the body. And that's one of the huge benefits of using mRNA. You inject it and it's gone. And in fact, if you had an mRNA vaccine, it was probably degraded before you got home from the pharmacist. And the protein was there and present for much longer. And we're using that to our advantage in some of these gene editing technologies like CRISPR, where we're introducing the gene editing vehicle as an mRNA. And the reason for that is, again, it goes away. And so you can make a gene editing mRNA, introduce it into the body, it'll do its action, and then it'll simply dissipate and be done. Where if you introduced it through other means, such as a DNA or a virus or something, then it could continue to do its action over and over again and maybe have adverse effects. So mRNA is really the way to go, I think, with a lot of these gene editing technologies because of its natural propensity to just be degraded.
Yeah, and just to drive home a point with that, so mRNA is just this incredibly powerful technology for medicine. And so in its standard way of using it, you're not doing any genetic change to the individual. You're just inducing a protein for a short amount of time and the mRNA goes away. If you do want to make the permanent change, though, with gene editing, there's a lot of technologies to use, but mRNA, because of these reasons Jeff just described, it is a very good approach to take. So it could enable gene editing, or it can be used in a very different way that doesn't change anything with genes in a permanent way.
This is a question for Dr. Jaffee specifically. Is there a concern that your research grant for developing the pancreatic cancer vaccine could be stopped? And if it was, would you have to end the clinical trial?
The answer is we are very concerned, and our patients are concerned. And yes, we don't have a lot of funding for doing these trials beyond the National Institute of Health. So, and the way things are going, I have to be honest that you're told a day in advance that your grant is cut. And so it will be an acute change for patients who are currently being treated in our clinical trials.
I think that might be it for questions. Thank you so much to our speakers and to everyone in the audience who took time out of a busy news cycle to hear more about this important work. Thank you.